Just video is fine. Speaker should be visible now.
The speaker is audible.
Video is fine. It's okay. Shall we any slides? Where are the other thing? Okay. So shall we? As the recording is everything's fine. Okay. Good. So I welcome you all for today's talk, and we have with us Professor Utpal Nath, one of my very good friends, and he works in the Department of Microbiology and Cell Biology of our institute, IISc. A little bit about his past. He did his BSc in Kalyani, Krishna Vidyalaya, right?
Vedanta Krishi Vishya.
Krishi Vidyalaya.
It's an agricultural university.
Right. Then followed by his MSc in Biotechnology at Tamil Nadu Agri University, Coimbatore, and then his PhD from 1991 to 1996 at NCBSKIFR. There is another excellence center in Bangalore, from where he is learning, and followed by his postdoc from the same place, and as well as John Innes Center, Norwich. And then he joined the institute in January 2003, and now he holds the position of a professor. His main research area is in organ growth and morphogenesis in plants, specifically Arabidopsis plant, and a lot of awards that he got. I will just name. He is the fellow of all three academies, which is a very, very hard thing to have. He got a whole lot of grants from DBT, DST, CSIR, which is another kind of recognition that he has. Recently, he has a very good paper in Nature Plants, which is supposed to be the best journal in plants, and one of the very celebrated ones. So, we are very happy and fortunate to have Professor Utpal Nath for our lab research program, and he kindly agreed to give a talk. I'll just hand it over to him so that we can enjoy the talk.
Thanks very much. I would first would like to thank Professor Mandal. I'll call him Partho as I call him. The very good friend of mine and colleague as well. I just want to know. So what I want to do today is I'll tell you what I work on. Okay, and I find it interesting. That's why I work on it, and I'll try to convince that is interesting to you guys. You can decide. It doesn't matter whether it's plant or not, because the question that you are addressing, which is the main thing in science, right? You have to address a question, right? And should have a very good approach to address the question, because the approach can change or maybe technically impossible or whatever. So you have to have some allergic cough, so that will come as free. It doesn't matter whether it's plant or not. The shape is onwards, right? You can, if you talk to an engineer, if you see an engineer at work, you can see that he's making some building or some structure which is a specific shape. But there is a design in the head of the engineer. But whatever is happening in the nature in front of you, whether it's plant or a baby growing or whatever, that is growing by itself. There's no design there. Right? If there is any design, it's within itself, right? In the organism, all the information is there, and is growing to a very specific way. That's why every human being looks very similar, right? So how that is regulated, how the growth is regulated, and how the regulation is such that each and every organism or organ within an organism grows to pretty much similar shape and size. Right? Like this is a limb, right? Or head, right? So on and so forth. So I'll try to see how that happens. In plant, you can address the same question. Animal as well. So I just wanted to ask you that at what stage of your academic career you are in? You are in BSc, MSc, MSc. All of you are masters. BSc. Okay, good. So that's even better. So I'll tell you how the leaf shape. I'll take leaf as an example. You can take Drosophila wing also, right? Insect wing also pretty similar to to a leaf. So I'll tell you how that happens. So to begin with, I'll tell you that this is a tiny plant of some species which is called snapdragon, and it is making leaf. Okay. So this is the first pair of leaf which came first. You see, it is attached here, and after that, the stem has grown so much. Okay. And then after probably a day or two, another pair of leaf came. Right? This is younger. Then after that is another pair of leaf appear. Then there is another one which you can't see, but is there. I mean, if you remove the older one, you can see. Then there is the fifth one also. The sixth one also is there, but you can't see. Why don't you see? They're too small. They're baby. Okay. So to see them, you have to remove all this rubbish and go all the way to the top and take a close-up photograph or maybe scanning electromicrograph or something and see how they look. It is a different problem that how they are nicely organized at one line here, and this is orthogonal to that, this is orthogonal to that. That arrangement is something I'm not going to talk about, but you can ask that question. How is it that in some plant, the leaves are like opposite? In another one, or two does not come, doesn't come as a pair. It's one here, one here, one here, one here, and it goes as a spiral. And the angle between the two actually follows the Fibonacci series or the golden angle. You know what is golden ratio and Fibonacci series, right? And if you don't know, you read it, you'll see it's very interesting. So, if you want to see that, what you will see is all the way down here. Where are they coming from? Are they appearing from this leaf appear from this tissue which is here? No, they are all coming from the same origin, a bunch of cells. They're stem cells. So you need stem cell for new organogenesis. How do I see the stem cell? So remove all this rubbish and go all the way to the top and take a scanning electrophotogram. Okay. I may change the species. Okay, the earlier was Anthracnum, this is Arabidopsis, but doesn't matter. All the plants are doing the same thing. Okay. So here, you see, this is the leaf, 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 this is the leaf. What is the stem cell here? The remaining part. This is called meristem. The stem cells are the one which will never ever get converted to any organ. They will always divide and stay what they are. No change in identity. But make some new cells and push it towards the periphery. Once they go to the periphery, they take the leaf identity. Now, leaf is very different. Okay, leaf will have a very specific cell type and function and so on and so forth, which is called morphogenesis, a differentiation. So this is actually primordium, which is the oldest one that you can see here. Now it doesn't look like leaf at all, right? It looks like a pav, no? The bread, pav bhaji pav. Yeah, it looks like a mound of a loaf of bread. This is the later one. 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 This is the youngest one that you can see here. Another one will come sometime here. This is what I was saying. So in this species, it is actually spiral. And it follows an angle which is roughly 138-degree angle, which is exactly at the Fibonacci angle, as they call it, or golden angle. I'm not talking about it. What I'm talking about is how this originates from here and grows to the mature leaf that I showed you already. Right? It is not mature, but still it's much bigger. Here I'm talking about only 50 micron long. Imagine micron, 0.05 millimeter, and the mature leaf is like this big. So it's growing dramatically. How does it grow? I want to see the rule. How does it grow? This stem is growing in this axis, and leaves are coming at the orthogonal axis. Just like here, this is the main axis, this is the orthogonal axis, so 90 axis. So how does it happen? How does this one, which is here, grows to this one? And every time you see the shape, it's pretty similar, right? Little bit difference is there, but by and large, it's similar. How does it happen? Where are you from? From which place? From Mysore. You came today or? How did you come? Train or? No, if I talk to you, you'll wake up. Okay, you won't go back to sleep. That's right. So it's good to sleep, but it's just that I'm jealous that I can't do it and you can do it. So that's why I hope you know. So see, this is the youngest leaf that you can see, 2 millimeter. Okay, I'm talking about the growth at a later part, not that 50 micron, 100 micron, 200 micron. Okay, because you can't see them, you have to do SEM every time. So this is a leaf which is very baby leaf. I just took it out and took a photograph. Next one, next one, next one, next one, next one, next one. So this is, you can imagine a leaf which is growing as a function of time. And this entire process takes about 12 to 15 years. After it is mature, no change will happen. But the leaf will stay on the plant for another 2 months and do its job. It has to do some job. I'm not actually interested in this one as of now. My interest is from the beginning till this maturity, change is happening in its shape and size. How is that happening? What is the rule of that growth? That every time every leaf looks very similar. Right? That's what I want to know. Like I've given some, see 2 millimeter long, 10 millimeter long, 35 millimeter long, 54, 70. So what you see here very readily is size is changing dramatically, yes or no? Right? Shape is not really changing much. You see, this tiny leaf is pretty much similar to the leaf which is here. So shape doesn't change, size increases. How do things happen? What is the rule, mathematical rule? Something which is shape doesn't change, is the size is increasing. Your intuitive answer will be that it is growing uniformly, isometrically. Okay, this region is growing at the same rate, and this region is growing at the same rate as this region. Then shape will not change. If one side grows more than the other, shape will immediately change. But shape is not changing. So your intuitive answer might be, my answer was, that prediction is every region will grow at the same rate. How do you know every region is growing at the same rate? How do you know this region grows at the same rate or not? There is no difference between, sorry? No, that is true, but that is a prediction. I'm saying, how do you do the experiment to show? See, prediction is good in the beginning with, right? It takes a fraction of a second. But then you have to actually do the experiment and show that my prediction is correct or not, right? Your prediction is correct, it is called null hypothesis. Opposite is alternative hypothesis. The H0 and H1 you would have done at least here. So there is no marker here or here. Is there a marker which looks very different from the base? No, right? So you can actually put some marker outside. Very simple experiment. Put some dot on the leaf and then see how the dots are going away from each other. Tell me, if it grows uniformly, then how do you think these dots will be at the end of the growth? The same position, not same position, relative positions. They will go away from each other. See, you have a balloon, okay, where there are spots. If you start inflating the balloon, don't you think the spots will go away from each other? Right? If it is going uniformly, balloon expanding, then the spots will go, the relative position will remain same, right? Distance will increase. So it will be something like that, isn't it? Right? That is another possibility. You can do this experiment in many plants. Another possibility. I would say that, look, what you are saying is possible, but what I am saying is that the center is not growing at all, only the periphery growth. All the peripheral cells is expanding. No growth in the center. How do you think the dots will be in the end? They will not move. Because in the periphery there is no dots, right? Yes. I take a rubber band. I expand it. Do you think only the end grew or entire region grew? Entire region grew. How do you know? That's not your guess. But you have to show me and convince me.
Some reference.
Put some dots. Put some dots in the center. No? Some 10 dots, equidistant. You expand it, you see the dots going away from each other everywhere. Right? But that's not how the fungal hypha grows. Have you seen fungal hypha under the microscope? One single linear structure. How does it grow? The entire hypha doesn't grow, only the tip grows. New material is added and tip grows. Just like a pillar is, the engineer makes a pillar. Add more, height increases. So add at the top. Right? That's how fungal hypha grows. So similarly, that is in one dimension, but this is in 2 dimensions. I can say the periphery grows. If the periphery grows, then the dots will not go away from each other. This is only to explain the approach of science. Okay, it's a very simple science, but at least you can get a nice answer. Okay, so you do the experiment. No? That was a cartoon that I showed. You do the experiment. This is a actual leaf which is 13 millimeter long, still growing, attached to the plant. In Adobe Photoshop, I have removed it. And this is the and I put some nice dots under the microscope. It will still grow. I showed you that is about 60, 70 millimeter long, right? This is only 13 millimeter. A lot of growth is still remaining in its life, right? Let it grow. The same leaf, when it grows, I took a photograph. This one. Where are the dots? Is it all over? It didn't really grow throughout, isn't it? So if you can't see this, I can see that is too small. So I'll normalize it. So this is the small leaf, this is the big leaf. Basic dreams, huh? So where does it grow from? It is not uniform growth, right? You see this one, which is here, has come here. See, all these dots are here, but all the dots are only in the top half, not in the bottom. Where is the growth from? Where is the growth from? More base, exactly. So it actually growing from the base. The top is not growing or growing less. It's not that enough. You see that the dots are crowded here, but all these dots are far away from each other. There's no dot here. So this 50% of this growth came from this roughly 10% of the area that there are no dots, because I couldn't go all the way down here. Otherwise, I would have broken it. So it doesn't grow uniformly, even though one of our what do you call it? Propositions or possibilities were that it grows uniform, but it doesn't. Right? Okay. So it doesn't grow from the growth. So it has a gradient of growth, maximum growth here, minimum growth here. Right? Or growth arrest, if you see, initially everything was growing, okay. Arrest has opposite definition. This is arrested most, this is less, little less, little less, less, no growth arrest, so growth is different. It's just the two sides of the coin. Yeah. Okay. Now, most of the growth is actually contributed by the cell division. Okay. Cell becomes 1, 1 to 2, 2 to 4, 4 to 8, 8 to 16, right? Like that, exponentially. So then, if the growth is happening here, the cell division also should be more here than less here. Yes or no? Right? And when cells divide, they don't really, individual cell does not become very big. They just divide into 2, grow a little bit, then 4, grow a little bit, then 8, grow a little bit. Cell remains small, but the number increases. That is the way of growth in an order. So check whether these cells are young and these cells are old or not. So how can you do it in a small leaf? You can't see the cells. So you increase your eye power using a scanning electron micrograph. So this is a scanning electron micrograph of this region, and this region. You see the average cell size is about 100 square micron. Area I'm talking about. I mean, ignoring the depth of the cell. Okay. So the two dimensions. The average cell area here is 1,500 square. The average area here is roughly 5. So you can see the cells are dividing more here, less here, and least here. Also, the differentiation, which is opposite of cell division, that means it will not divide anymore, is more here because you can see the stomata differentiated. No stomata here. These are stomata in the process of differentiating. So you can say that there is a gradient of growth, and these cells are actually dividing not only by size. There is a gene which is, what is gene? What is protein? There is a gene or its product protein is required for cell division. Where do you think will be more? At the base, because that's where the cell division is happening. Less and there will be least. So there should be a gradient of the gene expression, more here, most here, less here, and least here. Right? If it is related to cell division. Can you name one gene which is related to cell division? Only expressed during cell division, not later. Required for the cell division. If that gene is not there, cells won't divide. Cells divide by cycling, no? There's a cycling process, isn't there? M phase, G1 phase, G2 phase, S phase, G2 phase, like that. So what gene is expressed during the cell division and not later? What? Cells are cycling. Well, I'm trying to give you a clue. There's a gene called cyclin, CYCLIN. Is expressed only during the cell division, right? So if you do a cycling assay where they are expressing, and I won't spend time in telling how this experiment was done, wherever there is blue color, you take it granted that the cyclin is more. Okay? You see, there's a leaf which is only 3.2 millimeter long, 3.2 millimeter, really tiny leaf. You see, the cyclin is maximum here, minimum here, even less here, and there is no cyclin. So gradient is there for a gene which is required in the cell division. And if you take a younger one, cyclin is almost everywhere. If you take it even younger, it will be like equal. So initially when the leaf is growing, it is equal growth. Cell division everywhere. During the process, it is this region which continues to grow for the longest time and contributes to it. Okay. And little later you see it's almost gone, and then after some time when it is 10 millimeter long, probably it will completely go. There is no cell division at all. The rest of the growth will happen by individual cell expansion, but not by cell division. So this is how leaves grow. When the leaves grow, there is a gradient. That we have seen by putting some dot on it, that we have seen by cellular behavior, how big the cells are, that we have seen at the molecular level, also by cell division. Right? Okay. Now, if one region of an organ grows more than another region, this traditionally, by botanists and zoologists, and neither of them, is they call it allometry. Okay, what do you think allometry means? Allo means what? Okay, tell me opposite of allo is what? Iso. Iso means what? Same, equal. Allo means not same. Okay, metry means what? Metry? Metric system? Measurement. So allometry means opposite. It measures differently. Okay, that's called allometry. What is allometry? The definition of allometry is that growth relationship of one organ to the rest of the body. Like when I was growing, you measure my height and the length of my length and length of the hand, and see whether there is a correlation. Is it that the hand is growing fast and only my body grows? Do you think that's what happens? You don't see any baby who has a long hand and the baby has grown. There is a correlation of the growth. That correlation is actually called allometry. And this allometry, I will give you one example. The best example I know, this guy actually measured and mathematically expressed what should be the growth. So what he did is, I'm giving an example. So this is a baby who is growing. Okay, 8-week-old, really, really small. Inside mother, tiny. And little bigger, and she's born here, and here she is mature. Okay, I have normalized the height. Can you see the head? How much? So, if head here is almost 50%, isn't it? Yeah, of the total height. Whereas head here is about 12 to 15%. What does it mean? Our head becomes smaller as you grow? No, proportion of the head becomes smaller. That means our body grows more than our head grows. Because head has grown earlier. Yeah? Early growth of head and later head. So what he did is, he measured this one and plotted them one against the other. It's not a kinetic graph. It is one measurement as opposed to the measurement of the other. You see head here and trunk here. You see, it's not a linear graph. It should not be, because one is growing more than the other. So it should not be linear. It's a non-linear one. Have you studied any non-linear mathematics? Okay. No. Okay. Very good. Welcome aboard, because I also have to study. But this is not linear, that much you know. You know what is the mathematical equation for straight line? Y equal to mx plus c, right? But this is not y equal to mx plus c. What is it? This is called power law. So power law means that if it means y proportional to not x. If y proportional to x means it is straight line. It's not x. It's x to the power some value which is not 1. If it is 1, it's a straight line. It's either more than 1 or less than 1, but not 1. Okay, this is called power law. Alpha can be 2, 3, 4, 5. What does it mean? If alpha is 2, that means when x becomes double, y becomes 2 to the power 2, 4 times. If alpha is 3, then when x becomes double, y will be 2 to the power 3, that means 8 times. Yeah. So that is the power law. Okay, but you can convert this one into straight line, because I myself don't understand nonlinear. So if there's something nonlinear, I can convert into linear. How do you convert this into linear? Constant. Who said log? Exactly. You take log on both sides, right? You take log y. Okay, this is equal to some constant into this one that we put that, and you take the log on both sides. So log. It becomes a straight line. Y equal to mx plus c. This is straight line, right? So now, what does this alpha mean? Alpha means the tan theta of this straight line, or the slope of the straight line. What does slope means? For unit increase in head, how much trunk is increasing? That is what that alpha means. Okay, alpha is very interesting. parameter because it also tells you the directionality of the growth. How? Okay, here it is. This is the baby growing, this is a she has grown to adulthood. So when alpha is more than 1, here, alpha is more than 1. You know what alpha is, right? Y equal to x square alpha. When alpha is more than 1, it is called positive elementary. For human, it is 2.6. That means when my head grew twice, my body grew 2 to the power 2.6. Yeah, remember that. So when alpha is more than 1, it's called positive elementary. It means that there is a growth gradient in this direction. There is more growth here and less growth here. When alpha is less than 1, the gradient is opposite. It's called negative elementary. Remember, alpha is not negative. The allometry is negative. Alpha is less than 1, 0.6, 0.5, 0.7, so on and so forth. And when alpha equal to 1 is isometry, there is no allometry at all, right? The head grows twice, and the body grows twice. That doesn't happen for us. So this is the growth, that the way we grow. There is a mathematical relationship for everybody, not only for us, even for a crab also, even for cattle, for everything. This is the allometry is followed. So this isometry, there is no. I can stop anytime. So you tell me, because I have a reputation of talking too much. So and I have a lot of things to talk. So tell me when to stop, or the other partners will tell me when to stop. So this is the way it grows. Okay, that's what I have shown. Now all the leaves grow like that. Every human being grows like this. The what do I? Yes, every human being grows like that. Otherwise, our look will be very weird. Imagine our head is going twice and body is going twice. Go and put some water on your face. So you have to measure alpha value. Okay. In human, it is very easy because there is a head and there is a trunk. You know where it is? You know where water is? Hello, lady. So you can measure because I know that this is my head, but the leaf doesn't know where. I mean, you also don't know where is the head of the leaf, no? Because there isn't any head. How do I do the same thing here? Well, there are always ways to do it. What you can do is, here it is very easy. This is the head and this is trunk. Here, what you do is, when the leaf is still growing very small, we put a dot in the center and say that this is the head of the leaf and this is the trunk of the leaf. X and Y, right? Just same thing. You can do that on day 1, x equal to y. Isn't that correct? Yeah. And in the end, we already know that we grew more than this one. So y is greater than x, right? How do I do that for a leaf? This is how we do it for a leaf. You can now measure alpha value. How will you measure the alpha value? You tell me, how do you measure the alpha value? You tell me, how do you measure the alpha value for this leaf growth? Tiny leaf, big leaf. I just normalize. Plot what? Where are my 2 sets of data? Measure the x and y every day, right? As they grow, right? And have some set of data. Day 1, you will have x equal to y. It's okay, but you will get x1, y1. Okay. Day 2, x2, y2, x3, y3, x4, y4. You will have a set of data, and then you plot it in log plot. Take the tan theta, right? You will get alpha value. So you will know when this grew twice, how much this grew. Maybe 8 times or whatever it is. You don't know. So you do that for many leaves. These are the leaves that grow on our campus. You can do in your campus. Mysore campus, so absent leaf. A lot of plants are there. You can do that and see whether all the leaves grow the same way. There is no reason why all the leaves should grow. Or maybe yes, all the leaves should grow there. So you can measure the alpha value for all the leaves. Okay, I'm showing you a few. There is a bunch of plants, including tobacco, snapdragon I showed you, Bonfrena, this is balsam, and this is balsam, and so on and so forth. For all of them, this is the alpha value here, or growth ratio. This is 1, 2, 3, 4. You see, all of them alpha value is more than 1. Right? As long as it's more than 1, I will call them as positive allometry. And it is positive allometry, right? That we have seen. So we call it positive allometry. Big deal, I've already explained. But not all the plants are like that. There is a so what is your name? Pooja. City. So what I explained just now is that alpha value calculation you know how to calculate x and y, and you measure every day x1, y1, and plot them into log plot. The slope will be alpha value. Okay. You measure that for many plants, like these are the 75 80 species. Measure that for every plant. Many plants have positive alpha value because alpha is more than 1. But there is a large number of plants where alpha value is statistically. I am showing you the standard error bar also, no? Yeah. Standard deviation. It is 1. What does it mean? Isometry. There is no allometry here. The base is growing twice, is also growing twice. So I call it isometry. Alpha is more or less 1, and that's the value that I have shown. But the interesting is there are a few species where alpha value is statistically less than 1. What do you call it? Negative allometry. And what does it mean? Where is the growth more? At the base or at the top? At the top. It's very funny. Some are growing from the base, some are growing from the top. And some don't have any growth polarity at all. They're growing uniformly. There is one plant which is very weird, and I'll tell you later. I call it complex allometry. It is hidden here, or positive and negative allometry. Alpha is, I'll tell you what it is. It's not very easy. So when you put all of them together, you see almost 50% is positive allometry, all right, which I explained in the beginning. But there are a large number of percentage which is negative allometry. So there is a diversity. There is a divergence of allometry, and in nature there is a diversity of. Negative allometry is also there, isometry, mixed allometry, I explained that we are the one positive allometry. So let's look at it a little closer and then see whether there really cells are also growing, dividing more here in this one positive allometry and more at the top in the negative allometry. Let's have a little closer look. So to have a closer look, I can't do for all the 40 species, right? Too much. You take some representative species here. Suppose tobacco and tecoma. Tecoma is there, yellow flower. Hibiscus, which is there in the garden, and bohemia, that sunapata or cancel or whatever you call it. And you take it podium, which is the croton in your garden, is or dillenia, which is in front of envy. There is one big round fruit. That's why it's called elephant apple. Negative allometry, and takes one example of mixed allometry. And study them in detail. You see, young tobacco leaf, x equal to 1 day 1. And at the end of the growth, or when it grew, y equal to more than x. Right? This is what I have already explained to you. This is what I also grew. And you are not different. You also grew like that. And that's why I look like that. Okay? If I didn't grow like that, I'll show you how I would have grown. So alpha value here is 2.6. That's Newman alpha value. Here, I told you that it should be more than 1, which is 4.4. So when the top tiny tobacco leaf, if you take the top half, when it grows twice, at that time, the bottom half grows 2 to the power 2.4, 4.4. Huge, like 16 times more. Interesting growth. So what do you think the cell division should happen? Quick. Where do you think there will be more cell division? Base or tip? Base, right? And that's what happens. You see the cells here. Here, cells here and cells here. It's all mature. Yeah. Take section here, section here, section here. And stain the nuclei. Each nuclei means how many cells are there. You see, so many cells are there here because they're small. Crowded is somewhere in between. And if you look at the nuclei very closely, this is the experiment you would have done in your class 2 or in BSc. You can stain the nuclei and then see the cell division. Mitosis, prophase, metaphase, anaphase, telophase. Some of them will be like that. Anaphase, right? Count the number of nuclei that is in the M phase, mitotic phase. Express that as percentage of total cell. That is called mitotic index. The percentage of the cell that is undergoing mitosis. That's called mitotic index. Not that if you have 1,000 cells, not that all the cells are in the mitosis, isn't it? Some will be like G1, some will be G2, they'll be randomly distributed. So if you count that percentage of the cell here, you will see about 5.5% is mitotic. This is 2.5%. There is no cell which is mitotic. And the total count would have been about 1,000 here, here, and here. What does it mean? Lot of cells, at least 5.5 to 6% cells are. The cells are dividing more here, less here, and least here. Same thing that I showed you before, but in a different methodology. That was some transgenic plant with a reporter gene, and this is by analyzing the. So tobacco has positive allometry. You are convinced? Okay. I'll take another one, which is hibiscus. What is it? Isometry. Does it have any difference? Okay. Tiny tobacco leaf, all the dots are here. Bigger tobacco leaf, all the dots are here. It's not clear from the base or tip. So looks like it is growing without any other isometry. What will be the alpha value? 1, right? So you measure alpha value. This is x equal to y on the day 1, and this is the end of the experiment. Still x equal to y, right? So this is what I would have looked if I grew with isometry. Right? Head grew same as the thank God I did. And here, what do you think should be the alpha value? 1, 0.95 plus minus something which I am not showing here. I showed you before. Right? So statistically, it is 1. What do you think the cell division should be? More here, or here? Equal everywhere. You see the cell? Same size. Mitotic index. You see the crowding of the cell is pretty similar. This is the mitotic index here, and here. So synchronous growth. Whereas the earlier base was growing. Take another one. So this is isometry. Take another one. Podium, negative element. Take podium. The podium I'm taking because I'm sure it's there in your garden or in your university garden. Called proton. You see small proton, I think 40 millimeter long. This is, I think, gone to some 145 or something. Bigger one, just normalized. Day 1, x equal to y. End of the growth. You see, x is longer than y. Isn't it opposite of what you saw in tobacco? So tobacco, we saw the basal growth more here. Where do you think the cell division is? At the tip, right? So this group, what do you think the alpha value here? Yeah, exactly. So let proton grow. But as long as I don't grow it, because it would look very weird. No? This is what if I would have looked if it is negative. And look at that one, 0.6. Right? So it's negative element. The direction of the growth has now become upside down. And the cells, if you see these are bigger cells and these are smaller cells. That's what you would expect. Right? Mitotic index, 3.1% cells are in the M phase, 0.7%. No mitosis going on, no cell division going on. So it has. So now you have 3 different types of growth, right? At least for leaf. How does the stem grow, do you think? The stem grows like that, no? How do you think? Positive element and negative? It's always from the top. It does not have any diversity, right? It can't grow from here. This is an interview question. You go to a tree, okay? Put some 2 nails 1 foot away here and 1 foot away at the top. Go back after 10 years. Will the nails go away from each other? No here, but yes here. No, because this is the what is growing. Every tree grows with single leaf doesn't. Leaf has diversity. Okay, I told you that one weird thing, no? This one? Okay, example, sization. This one. Here, the cells are larger here, but smaller. What is the growth? In the middle. And you cannot measure alpha value because for alpha value, not only you need an axis, you need a unidirectional axis. Growth should happen in one direction. And here is happening one direction, here another direction. How do you measure the alpha value? Not possible. But possible. You can measure alpha value of this one separately and this one separately. And say that distal alpha value, proximal. Right? What do you think the alpha value will be here? Remember, more growth here, less growth here. What will be the alpha value? More than 1, right? Positive elementary. And here will be negative elementary because growth here more than that. You measure that separately. This is 1 alpha value. Where is it? This is 1 alpha value. This grows in the middle. Okay, good that I didn't grow from the middle. That's why I don't look. Here it is 2.7. I don't know, it's not coming here. This is 2.73 and this is about 0.6. So growth is from the middle. Okay. So this now I quickly go through. And now we have this kind of diversity. Growth more here, growth more here. There is no polarity at all. It is the bipolar. No, I mean I didn't mean that bipolar. The doctors talk about. I mean it has 2 directions of bidirectional. So now there for everything in a reason, no? All the leaves are, all the species that you see, one day they are all small and tiny and baby, and they are growing, but there are different directions. There might be some molecule or something which will be regulating that, right? For example, when they are growing, some gene which promotes growth may be more at the base with the positive element. Maybe the same gene is more at the tip when it is negative allometry, right? In another species. The same gene where there is no allometry at all, that means isometry, maybe it is equally distributed along the proximal axis. Right? That's one possibility. Or maybe it's not a growth gene which promotes growth, some gene which suppresses growth. In positive allometry, it is more towards the tip when the growth is arrested, where the growth is arrested. In negative allometry, it is more here. So some directional and unequal distribution of some molecule has to be there. Let's see what it is there. So you find out all the genes that are required for growth. People have done it for you, so you don't have to worry about it. You just read some paper and you will have a list of 20 genes. Some of them are promoting leaf growth. If you remove them, their function leaf will not grow. Some of them suppressing leaf. So one of them, of course, when you do all the screen and see their expression pattern in Arabidopsis or any species, you don't show all the data. One of them you can see that. See, what you are saying is baby Arabidopsis leaf, seed leaf, and you are seeing from the top. Okay. You see this, whenever blue color, this GRF or growth regulating factor is more, right? More the blue color, more is the. So you see in this leaf, it is more here, less here, less here, less here, and there is no expression. So there is a gradient here. So one possibility is that this GRF gene is expressing here more because growth is more here. Arabidopsis has positive allometry, by the way. Okay. In a negative allometry, maybe the same gene in another species is expressing more here. You can isolate the GRF gene from Arabidopsis, which is already somebody else done. I can request and get that gene sequence. Okay. You can do that for other species, nobody has worked. So how will you find out the GRF gene from hibiscus? Nobody has isolated. You can isolate. But the problem is there are 8 GRF genes in Arabidopsis itself. And in other species, there are 20, 10 and all, because important genes come as a family. Not all of them probably do the same thing. But so you can't do that. But thankfully, there is another gene which is not promoting growth, but suppressing the GRF. It's a microRNA. You know what is microRNA? Very tiny RNA, 20, 21 nucleotide. It's a regulatory RNA. Have you heard about microRNA? No? miRNA? No? You should read this one. This is one discovery which got a Nobel Prize within 3 years because it is so dramatically, exciting. There are certain small RNA which suppress other mRNAs. It degrades because it is attached to some RNAs. It goes along and binds to the mRNA, its target mRNA, and degrades the target. So, for our growth and development and functioning, genes should be expressed. But it is also true that genes should not be expressed. It should be repressed. Right? Every gene is expressing all the time. That's a nightmare, isn't it? I gene is expressing in the liver. Terrible. So there should be some regulation. This is the regulatory RNA. How does it bind? Which RNA? There are 10,000 mRNAs encoding 10,000 proteins. Which the microRNA should bind to which mRNA? How is it regulated? But the sequence complementary. microRNA, each microRNA has a specific sequence. If that has a complementary to the mRNA, it will go and bind and then cut and it will degrade. Okay. So this microRNA is known to degrade these GRF genes. And it is expression pattern is exactly opposite. It is more here and this. So I'm telling you that my hypothesis is that this mRNA is regulating this diversity in growth. Why do you think this mRNA should be more here or here? Very good, because it's a maturity inducing RNA or growth arresting RNA. Will be more here, here, at the base here, equally distributed here, maybe more here, but less here. Let's see whether it is or not. It's not very difficult. Well, it's not very easy either. So what you do is, you go to a young leaf, pluck it, cut it into half, and then see whether microRNA is more here or less. That's all, right? So I'll tell you what was done. This is a tekomma leaf. Okay, you cut into half. You see that cells are bigger here and smaller here. That means the growth must be here. This is a positive allometry. So you can see this gradient that tells you. There is another one which has negative allometry. Proton, more here, growth, and less here. Okay. There is another one which is Sonapata, Bohemian, Kanchan. It has no. Why am I showing Sonapata here and not the hibiscus? It's nightmare to isolate RNA and DNA from some species. You know what you get? Just like the phlegm when you have cold, that like sticky thing comes out because plants have so much of carbohydrate that it interferes with the nucleic acid isolation. So you try some 10 different species wherever you get RNA. So this gives nice RNA. What you do is you just cut into top and base, top and base, and top and base. Okay? Mash it up. All the small RNA you run in a gel. You know what is that gel, agarose gel? You have tons of RNA, like probably 500 RNA of different sizes and all. You run them in an electrical field, depending on the size, it will get separated. If it's a big one, then it will get stuck because it can't move through a crowd. If it is a small one, it will move fast. That's how we separate based on this. So you do that of this entire RNA and entire RNA here in a gel, and then see where is your RNA, that specific RNA, by something called northern hybridization. Heard about northern hybridization? Yeah. Why is it northern?
Because it's towards
That's why it is called northern. But if I turn the one toward the south, then it becomes southern. That's right. Why is it called northern? Where is the direction they come from? Different labs have different direction. It can't be that direction. Some guy in San Diego first showed this one for DNA fragment. The small DNA will run first in electrical field and big DNA. He devised a methodology where he can find out where your specific DNA is here. His name was Southern. S-O-U-T-H-E-R-A. Adla. Very directional name. So this is called Southern. Later, people found out that you can do that for RNA also. Little bit tweaking, or in the protocol, you can do that. What do I call it? Okay, Northern. Later, they found out you can do for protein also. Big protein will run later, and small protein will go through. Because it's like, a tiny boy can run through a crowd very easily, right? But someone will, will be difficult. So it's the same thing. So protein now to detect antibody, right? You can find out where with the hundreds of proteins, thousands of proteins, you can find out where your protein is. So you call it Western. There's no Eastern as yet, but that's how the names get from. So this is Northern. What you do is total RNA here from the chip, okay? Mash it up, run in a gel, separate out your specific RNA, which is 396, I told you. No? There are hundreds of them, no? They're separated. Your interest is 396. You want to know whether it's more here or more here. So you radioactively labeled your 396 tiny probe, actual RNA, add it there, and wherever your RNA is there, will go and bind there. Because your probe is complementary, you're taking, not the same. So it will bind there, radioactivity is there, you'll find it. You see at the tip, there's this much MIR 396 is here at the tip, but at the base is less. Right? So that means at least one, your prediction is correct. That in this species, the maturity mRNA is more at the tip and less at the tip. Yes? But you can tell me that, look, you cheated, which I am capable of doing. How did I cheat? I actually loaded more RNA here and less RNA here. Then also I'll see the same thing. So how do I know this specifically this RNA is more here and less here, or all the RNAs are more here and less here because I intentionally added more? Right? What do? You take another small RNA, repeat the experiment, load the same amount of RNA here and here, but you take another RNA, U6 mRNA, U6 RNA. I don't know there is no U6 RNA. You don't know what is U6 RNA? You guys are not reading textbook. Which molecular biology textbook you write? You read? Molecular biology. Tell me. You don't read. Only notes. You are from mathematics department. Anybody from biology? No. Which you are BSc or MSc? No, I'm not talking about microbiology, molecular biology. Nothing you have read is all YouTube. Your entire study is YouTube based, or is it textbook also? You should read textbook, okay? Molecular biology of the gene by Watson, the guy who solve DNA structure. Anyway, you should read that. So what I will say. Now, U6RNA is required for splicing, mRNA splicing. Okay, that. So is another small RNA is equally distributed. That means I have taken same amount of RNA, total RNA. This RNA specifically is more here and less here. Okay. Right. You see here, this is the tip and this is the base. What is the distribution? Opposite, isn't it? More at the base, because we know that this leaf is growing from the tip. Here, this is isometric. You see, equally distributed. In all, the other RNA is equally distributed. That means loading is same. It's just that specifically this RNA is more here, and equally distributed. Okay. I think he's giving a signal that you stop. I don't know. So wherever there is more RNA, its target GRF should be more or less. This is microRNA which degrades the mRNA of its target. A target is GRF, I told you. Wherever there is more RNA, how do you think that the target mRNA should be distributed? More miRNA, which degrades mRNA, it should be less, no? mRNA. You see here, one of the target here. Wherever there is more miRNA, that its target is less because it degrades, I told you. Opposite distribution. Here also more, less, more, less. Equal. So not only miRNA is more and less, but its target also is getting separated. That's because miRNA is more and less. So the MIR 396 is distributed in many different ways. Like that, like this, the gradient, and there is no gradient at all. What about this one, this funny one, where there is maturity here, immaturity here? What will happen? So, no, you cut it into half, no. Then what will be the distribution? Equal, right? More, less, more. If you add them up, it's same. So you cut into three. This third, and this third. Right? Why 3/3? Because you can't more than 3/3. Anyway, when you say third, it has to be 3. So you do the northern. You see at the tip there is more microRNA, at the base there is more microRNA, but here it's less. You see the association? Wherever there is more growth, there is less microRNA. Wherever there is less growth, there is more microRNA. And even if you change the orientation, the direction and all, association is very clear. So can I now conclude that this microRNA is actually regulating this polarity of the group? Yes. Who said yes? How many of you say yes? 3. Others don't want to say or non-committal? May or may not be, or I don't care. See, it's only association. Don't say that this not yet. This mRNA is causing it. You know, cause and effect. Maybe it's effect. Maybe somebody else is doing it. The downstream effect is this microRNA also is changing. That's also possible. If I give you another, example where maybe growth is more here and less here, which I have not shown and I haven't found also. Then also you see that it is matching with the microRNA. Then also you can't say that is actually causal. You can say that is strongly associated. Right? That's what you get. So your conclusion in science, if you want to be a scientist, remember this one. Your conclusion of your experiment should be only as much as supported by the data. Don't conclude too much unless you're a mathematician. Why? Because if I have the data point like this and if I can plot it into a straight line, I can extend that because I have a formula in my hand, y equal to mx plus c. That's why I can expand. But you can't do that for all the experiments, okay? Biology or any other physics or chemistry. Only as much as your data. This I typically communicate this to you by telling a very small story which you may have heard. There's a physicist and there's a mathematician, there's a philosopher, all their friends. And they're traveling together in a train in Scotland. You can imagine, Harry Potter, Ron and Hermione are traveling together. And they look out and then see in a field there is a sheep which is black in color, which is very unusual. Sheep are not black. So the physicists say that, look, the sheep in Scotland are black in color. So the mathematician says, look friend, this is not a very correct conclusion. You are computing too much. You see only one. Others may not be that. How do you? The philosopher says, look, that also is not correct. The correct conclusion should be there is at least one field in Scotland which has at least one sheet whose at least one side is black. But you see the other side? No, right? If you see, you just say, oh, this is a black sheet. It's not a black sheet, it's half black sheet. You know how to know the other side. So, do you know what is the other side of the moon looks like? You did not know until the Russians sent a satellite, isn't it? Because you are always seeing one side of the moon. The other side maybe look very different, or you have no idea. So your conclusion should be as much as your data shows. So sorry. Okay, it's all up to you.
Yeah,
I can continue for another one.
No. I think.
And if you guys want to hear me for the second half of my story, then I can give you an online talk, so you all can attend. That's not a problem. Or maybe it is there in some YouTube, or maybe it's not there. I don't know. But I find these are very exciting. Oh, I see. Even battery knows that my talk is over. I give up. I haven't spoken about the shape part of it. I'm only talking about the growth rule. How organs grow. Yes.
Yeah. So I think what we can do, maybe we'll have the same link applies. It means that we'll have the same link, and so maybe after a couple of more lab visits, we can actually return to the next half.
I have no problem.
That would be really great to really continue this.
Online, offline, I don't mind. But
overall, I'm really impressed by the way things have been presented. It's a really great talk about. I mean, I know very less number of. I've learned whatever I have known about. The number of talks in plants are very limited. I don't know for what reason, but the research itself in plant is not that much as it is for animals or any other kind of things. So the point is, the talk that we listened just now is basically mathematically supported. I mean, that would be the best way to do science. When it's supported by maths. Achha. Right. So first of all, thanks a lot for right. And the thing is now about question answer. Anyone has some interesting questions? Really, very absorbing talk. So
when the leaf grows, it will roll up also. Sometimes it can roll up clockwise, anti-clockwise. So does it have any role?
Can you pluck a leaf and show me its rolling?
If it is not a leaf, then some stem, like it can get a spiral shape. That can be like.
It's not stem. It's tangent.
Okay. Yeah, tangent.
Yeah.
So
no, your question is very valid because leaves are rolled. Some leaves are rolled depending on what stage of growth you are talking about. Look at a banana plant.
Yeah.
Have you seen banana plants? When it comes out, if it is already flat, it can't come out, right? Because it's coming deep from inside. You know where banana plant a leaf is coming from? When you see it has already traveled this path. Where did it start from? Ground. And that's where new banana babies come out. No? So all the stem cell I talked about is not at the tip, it's at the base in banana. It has to travel from here till here. It can't travel if it's flat. So it stays rolled intention. That's because one side of the cell expansion has happened earlier than the inner side. Outer side has already happened. So it is rolled. When it comes out, then the inner cells finish their unfinished job. Now they become bigger and now it is flat. So if there is unequal growth in the dorsal side and ventral side, you can decide which one is dorsal, which one is ventral. Then the leaves will grow. Sometimes leaves are mature leaves I'm talking about are infected by virus. Okay, virus can fly and come. How do they do it? Some insects or mites they carry the virus, just like the dengue virus is carried by the mosquito virus. So it infects this side, brings something from this side, virus goes in and it modulates the growth. It stopped growth, but it now reinitiates growth. There's more growth here, leaves become growth. There are leaf roll viruses also. So yes, if there is unequal growth on the dorsal side and ventral side, then it bends this way. Yeah. So there are specific genes which are expressed in the dorsal side of the leaf and ventral side of the leaf separately. You injure them, leaf will go like that or like that, depending on where the growth is more or less. So things are not that simple. So many things are going on in the background. We don't know. Just take one unsuspecting leaf. You think it's just a mere leaf, but so many things are going on in terms of polarity, growth, and all to keep it the way it is.
Okay. Anyone else want to ask anything? Okay. Utpal, I have some questions which I found very interesting in your talk, basically. So, what decides the rate of growth in different parts during development? This is one part. Other is, do you have a specific value for alpha during growth? If this value is specific for a specific species. And the last part is, what decide positive and negative allotropy at fundamental level? What means, what prompts this?
Very good question. I will do some hand waving answer for the last one because I don't know the answer yet. First is what determines that the growth rate, okay, of different region during the development. Remember, when somebody is growing or some organ is growing, there are many parameters of growth. What are the parameters? One is growth rate. Okay. Growth rate means you take about 4 different dots and join them, you have a square. Okay. The square has some area. In T1 or time 1 is some area. T2 is 1 day later. It will be more than T1 because it's growing. Right? In the 3rd day there will be more. So you can draw the area growth as a function of time and you can find out what is the growth rate. Okay? That is growth rate. There is another one is called anisotropy. If a circle is there, if it grows uniformly, it will be bigger circle. If it grows uniformly, it will be even bigger circle. There is no anisometry here, all isometry. However, if the growth is more in one direction than another, then circle will quickly get converted into an ellipse, right? Because the growth is more in this side than in the orthogonal side. So that is called anisotropy. The third is the direction. Yes, I agree that one side is growing more than the orthogonal side. But is it in this direction, this direction, this direction? If it grows more in this direction than this, the leaf will be wider than longer, right? But if it grows like this, then it will be longer than that. These are 3 typical parameters of growth. He is asking about the rate of the growth. What determines the rate of the growth? The expression pattern of certain growth promoting factor, just like GRF. You saw that GRF is more here, less here, and least here. Remember, when the leaf was very small, the GRF was distributed perfectly normal, equal. Later on, because of the microRNA comes out here, up here, it actually degrades this one. So it's an interplay of these growth promoting and growth regulating gene. And finally, you get the resultant growth rate is the function of those multiple resultant function. That's one. Second is, third one I remember because it's more interesting. But what is the second one? Which is also interesting. Oh, I don't have to answer. He has forgotten. Oh, hi. He has forgotten the question.
So, do we have a specific value for alpha?
Right. Is it species specific? Right.
Yes.
Alpha value is species specific. And if you measure, not only, so we have done for a specific species, okay? We have done it in IISc, we have done in BHU. The student who did the job is the MSc did in BHU, in zoology, and then we did worked in plant here. We did it in Coimbatore also. That's where I did my MSc, so I was visiting for some other reason. So it is depending on where geographical area, it doesn't matter. Same positive. So it is specific to species. We also did the leaves which is inside the tree and outside the tree. This is getting more resources, right? Inside is getting less. Alpha remains same. We actually did that I told you that put the dot in the center. Why should I put in the center? I can put in the 25% from the tip, no? Right? And then measure the growth here and as a function of time. Then also it remains same. So once it's positive, it's always positive. So it is specific to a species.
So alpha could be a way to fingerprint the species.
Absolutely, you can do that. Alpha value. Remember, what I did is more than one, it is positive element. I don't care whether it is 2 or 4 or 8. Whereas what he's saying is you can actually find out the alpha value and mark that as a species. This is 2.75. This is this species. Whether you can do or not, I don't know, because alpha value may have a lot of noise. But positive is always positive. I've never seen that positive is changing. Always positive, but may vary. Or the leaf that is coming out early in leaf in plant's life may be a little bit different from which comes later. But positive is always positive. Your last question: why is it there? Okay. First question is who knows? Who cares? Okay, because why is a domain of philosophy. Why do you live? Why the organisms are there in the on earth? Who cares? Okay, you've got a life, live. Okay, that's all you can do. How it does is something which is more the domain of science. Okay, but his question is very interesting. How does it happen? Well, by microRNA regulation and all, which I explained. That's how it happens. Why is it there? It's not that easy, the answer that I said, because the question is more than that. Now, the fact that the allometry is diverse means developmentally it is possible. Right? Diversity. It is possible that leaf grows from the top, leaf grows from the base, leaf grows from the middle, or whatever it is. There is no constraint, growth constraint. It can grow from the tip, grow from the base, because it is there. Right? Now, why is it there? We don't know, because there is no diversity, there is no constraint, it is there. Thousands of species are there. It so happened that in evolution, that's how it was selected. Now, we wanted to know that why is it there? Is there some link, some association with something? So we divided the plant, about 80 species, into different types. Okay, is there any botanist here? There's no botanist here. I'm not a botanist also. But I heard that there is some plants which are called annual. You know, annual? It grows for 3-4 months and dies. Rice, chili can be perennial. There are some which are perennial. Okay, year after year it will grow and become fatter and fatter and fatter. There are some plants which are called shrub, somewhere in between. So some plants grow at the ground level, some plants grow at the bush level, maybe giraffe also can. But there are trees which are beyond giraffe also. So there are different levels of it. Now, these plants which are growing here at the ground level, they are eaten up by animals, right? Thousands of millions of animals, grazing animals, which are every day eating lots of plants. Imagine if you have negative allometry of leaf growth, okay? Half of the leaf is eaten. That's the end of the leaf's life, isn't it? Because growth potential was here. It will stop here. However, if you are growing from the base, even if you take the top half, there's no problem. The young leaf is, growth potential is here. So what we did is, we did some experiment where very early on in tobacco, we cut the top half of the leaf. Very tiny leaf. It grew to normal size. There's no problem. We did the same thing for croton. You do that experiment on IISc campus. Tiny croton plant, cut the top half, the leaf dies. Doesn't grow. So, and we saw that out of the 80 species, 25 was annual. All annual are positive allometry. All animals. However, in the trees, it has positive allometry, negative allometry, and asymmetry, everything. So, I can try to answer your question. Is no constraint of growth in growing with all kind of diverse allometry, hence it is there. Okay.
Does this as a evolutionary advantage?
So that's what I'm coming to. Yes. So second, see, animals came much later. Plants have been there in the beginning of the evolution, and animals apparently came much later. Okay. When the animals appeared after the dinosaur, right? That's when the mammals, took over. They started grazing the plant like no tomorrow. Okay, all over the. Now you keep, and what will be eaten first, tree or animals? Animals grazing grass. Okay, you mow your lawn. Do you mow your lawn once in your lifetime? Every month you have to go and mow your lawn, right? Why do you do that? Because growth potential in the base is negative element, a positive element. So you cut here, it grows again. Cut here, it grows again. Every month, every 2 months you have to go. So evolutionary advantage is this, that initially when the grazing animals are not there, probably animals also had positive elementary, negative elementary, whatever it is, all sorts of because there is no constraint. However, when the grazing animals started, the species which is growing from the tip are already wiped out, right? Because they have adaptive disadvantage, and the plants with the negative, positive elementary, the smaller plants, shorter plants, they have adaptive advantage because tip is eaten up, the base will grow like grass. Right. So one possibility is that positive elementary has an adaptive advantage and survival advantage. If it is the grazing or eating up pressure is there, that's a selection pressure. Whereas the trees don't have any selection pressure, so they have everything. So one way of answering your.
Good. Anyone else has anything to ask? Yeah.
See, all the species have some allometer values. That is possible to change with the structure, DNA structure.
So actually, this is a important question, and I wanted to follow it up, but I didn't put that data because it becomes too much. I told you that Scottish story, no? Yes. So that is only association. Your conclusion should be as much as the data shows. Now, I have shown association of the expression domain of microRNA and growth inhibition. Okay. How do you know this is actually causal and not just effect? You can answer that, and we tried that. In Arabidopsis, which is the positive elementary, we saw there is more microRNA here than here. Okay. What we did is we took this microRNA and expressed that in a transgenic plant where under a regulatory region, where the regulatory region is more active here than here. There are many genes which are active here than here. See, gene has to. What is your background?
An engineer.
Okay, engineer, sir. I'll tell you. So gene has two major parts. One is the lower part is make some protein or something which are the like workers. They are the one who actually do the job, enzyme or something, or microRNA or something. The upper part actually is the regulatory part. That doesn't come into the protein. That determines where, when, and how much gene should be expressed. A gene can't be expressed all the time. No, I gene has to be expressed only in the eye. Right? So that regulatory part determines that. So that regulatory part, there are some genes whose regulatory part is such that there is more here and less here, like GRFI showed. Right? So I took a regulatory part of a gene which is expressed more here and less here. Okay? And took the microRNA and fused them. So now the microRNA will be expressed more here and less here. Right? So I changed that in Arabidopsis, elementary change. So this is not only is possible, it has been done, and you can change that. Only then you can say that is causal and not effect. Okay. So yes, your question is very valid, and we are there.
Alright. So I guess we can thank thank him for all the interesting question answers that we asked.
Oh, I forgot. Not only you. Okay, thanks for thanking. I'm taking that thanks, but my student also should thank me. Okay, I'm not showing that one because it's their work. I am only presenting on their behalf. So Manak Dasgupta, who actually did the most of the job that I have told today, and there are other students who did other work, which I hope that I'll tell later. But this part I missed. That I must thank the students, or rather they thank me. I don't know because work is theirs.
Okay, then we can actually wind up, and thanks a lot. Okay, so I think it's a lunch time.